Hello and welcome to my Othal teacher. Today we're going to talk about physiology of vision, color vision, and its anomalies. So, whenever there is light falling on the retina, there are two things happening: a photochemical reaction and an electrical reaction. This is what enables us to actually see. Now, rods and cones, as we know, are the photoreceptors of the retina, right? These rods and cones they absorb light energy in a particular spectrum of wavelength. For example, blue cone. Blue cones absorb blue light, which is of shorter wavelength, and this is the spectrum of the blue cones. Similarly, green cones absorb medium wavelength or green light, red cones, red light, and rods absorb light of the spectrum of about 500 nanometers. So, each of these photoreceptors have different absorption spectra of light. Now, I said that there is a photochemical and there is an electrical component happening when light falls on the retina. So, what is the photochemical reaction that is happening? That reaction is the rhodopsin cycle. Now, the rhodopsin cycle is basically a process which ultimately leads to the activation of the rods such that an electrical impulse is generated. It is that electrical impulse which travels to the brain. So, what is that chemical reaction happening that is responsible for activation of the rods? So, this is the rhodopsin cycle. You see, rhodopsin is the inactive form of the rod pigment. Okay, it is made up of two things: 11 cis retinal, which is a chromophore or the pigment part, and opsin, which is the protein part. A chromophore plus protein makes up the rod visual pigment, that is rhodopsin. A similar visual pigment is also present in the cones. Remember that. So, when there is light falling on the rods, activation of rhodopsin happens. Now, what actually is undergoing activation? It is the opsin. See, rhodopsin gets converted to beta-rhodopsin in a matter of picoseconds. PS stands for picoseconds. Then lumirhodopsin, microseconds, meta-rhodopsin, milliseconds, and meta-rhodopsin 2 in seconds. So you see the whole, activation as the light falls on the retina is happening in a matter of seconds. So it is the meta-rhodopsin 2 part of the rhodopsin which is the activated part. It is the activated rhodopsin and it gives rise to the nerve impulse. How does it do that? Activated rhodopsin. What is there in this activated rhodopsin is all transretinal. It is this all transretinal which generates the electric potential. Okay, all transretinal. So metarhodopsin 2 gives two things: all transretinal and scotopsin. The all transretinal is basically the chromophore that is responsible for generating electrical potential. And what happens in the dark is that scotopsin is regenerated. Okay, so this is in the dark. The regeneration of the rhodopsin happens with the help of scotopsin, and trans is converted back to its cis form, the inactive form. So scotopsin and the inactive form of all trans retinal, that is 11 cis retinal, in dark combine to regenerate the rhodopsin. This is what the rhodopsin cycle is all about. Now, for those of you who are wondering that what happens in the cones also must be having a visual cycle. So yes, cones use a different set of opsins. You know that opsins are proteins, so they just have different amino acid sequences. Light exposure causes a similar bleaching process in cone pigments. This results in an increase in retinyl esters, which generate the electrical impulse. So the process is similar. The opsins are structurally different. The retinal esters are formed, which generate the electrical impulse, and the whole process of you know cone visual pigment activation, that is bleaching, and their deactivation, reactivation, the whole process is just faster than the rod visual cycle. So now that that chemical component has formed and it has generated an electrical potential, what happens? What does that electrical potential do and how is it generated? That electrical potential that is generated in the rods or the cones is actually a hyperpolarization current. Now, unlike other photoreceptors of the body, the photoreceptors in the retina are in a state of, they are already in a state of depolarization. In other neural membranes, uh, activation actually leads to depolarization. But in photoreceptors, activation leads to hyperpolarization, that is a more negative potential. Now, why is this so? See, this is in the resting stage. For example, this is a rod. Now, there is constant leakage of sodium ions from the rod into the extracellular cellular space. There is a constant leakage of the sodium currents. But in order to maintain a balance, now it is because of the leakage of the sodium currents that the resting membrane potential is around minus 40 millivolts. Okay. Now, to maintain a balance, this leakage, there is a sodium pump also. So it keeps going in, coming out, going in and coming out, such that overall there is more sodium leaking out, leading to a resting membrane potential of negative, that is minus 40 millivolts. Now, what happens on activation is that the movement of sodium ions back into the rod outer segments, these are the outer segments, okay, back into the outer segments is decreased. So less of positive charge is moving inside. So the negative charge that was already there is becoming even more negative. Got it? This is known as hyperpolarization. So when activated, because of the leakage current, because of this decrease, the action potential becomes more negative, or the inside of the rod membrane becomes more negative, and that reaches to about minus 70 to minus 80 millivolts. And that is the action potential. All this is happening in the presence of light. The photochemical process has already happened. The rhodopsin has already happened. And what does that rhodopsin do? It leads to activation. What is this activation in the form of? It is in the form of further decrease in the resting membrane potential, leading to hyperpolarization and generation of an action potential of minus 70 to minus 80 millivolts. Now, the action potential is generated here, right? We talked about that. We talked about the rhodopsin cycle. We talked about how the action potential is generated. How does it travel? You see, this is the retina. This is the ultrastructure of the retina, a diagrammatic representation. So it begins here, the action potential or the current. Simply put, it travels to the bipolar cells, from there to the retinal ganglion cells. The retinal ganglion cells, their axons lead to the formation of optic nerve, which is a second order neuron. So here, let us look at what happens inside these ganglion cells. A little bit of detail. So there are two types of ganglion cells, midget and parasol. Okay, the midget cells, now from the ganglion cells, the optic nerve goes to the, we're talking about the visual pathway, right? So the ganglion cells, the optic nerve goes to the chiasma. From the chiasma, it goes to the optic tract and then to the lateral geniculate nucleus, right? That is the normal visual pathway. I hope everybody remembers. So it is the second order neuron or the ganglion cells only that are traveling all along the optic nerve, chiasma, and up to the lateral geniculate body, such that from the lateral geniculate body, the third order neurons emerge. So all of this pathway is the second order neurons only, right? Okay. So what are those second order neurons? These are the ganglion cells. Now again, back to what we were discussing, there are two types of ganglion cells, that is midget and parasol. What is the importance of these two types? The midget cells synapse in layers 3 to 6 of the lateral geniculate nucleus, forming the parvocellular pathway. And the parasol cells or the P cells synapse in the layers 1 to 2 of the lateral geniculate nucleus and form the magnocellular pathway. Now, these two pathways are responsible for different sensations. For example, parvocellular pathway has a sustained response. It sees color, poor in contrast, better spatial resolution, and is slower. Whereas magnocellular pathway has a transient response. It is monochrome, high contrast, low spatial resolution, and faster. So this is what these two different types of ganglion cells do. So from the ganglion cells, information goes to the visual cortex via lateral geniculate nucleus through the third order neurons. So the visual cortex is again primary visual cortex, that is the striate cortex, and secondary visual cortex, the cortex that is extrastriate cortex. Remember we talked about Brodmann's area when we read anatomy. So primary visual cortex is Brodmann's area 17 and secondary visual cortex is Brodmann's area 18, 19. So 17, 18 and 19 Brodmann's area ultimately form the visual cortex. Now, the lateral geniculate nucleus cells, as we have seen, are of magnocellular pathway and parvocellular pathway, which we saw in the previous slide. There are also conio cells. Okay, so lateral geniculate nucleus, striate cortex, extrastriate cortex, and higher centers. So ultimately, it is these magno cells and parvo cells only that are leading to these sensations, as I told you in the previous slide, that they lead to a particular type of sensations. That is why they exist. So magno cells are responsible for motion and stereo, that is 3D. Parvo cells are responsible for form, and conio cells are responsible for color. Right? So now the chemical reaction has happened. The electrical impulse is generated by hyperpolarization. It has traveled from the photoreceptors to the ganglion cells, to the lateral geniculate nucleus, and to the primary visual cortex. Now, at the level of the primary visual cortex, there are what we actually see. For example, we look at an object and it is the primary visual cortex where we process that object that we are seeing. For example, when we look at a flower, we appreciate its light, we appreciate the light coming from it, the form, what shape it is, the contrast of each of the petals of that flower, of the leaves, of the colors that are there in the flower, and the color of the flower itself. Any object is made up of these four components. I mean to say, the visual perception of any object is made up of appreciation of these four things: light, form, contrast, and color. Right? Now, you know that rods are more sensitive to low illumination than cones. These are just basic points. So during dusk, the rods are more active, and that is our scotopic vision. And in bright light, the cones are active, that is the photopic vision. And in twilight, both rods and cones are active, and that is the mesopic vision. Now, let's talk about each of these senses in detail. So, form sense. Visual acuity, when we say visual acuity, it is the capacity to see fine details of objects in the visual field and applies to central vision, which is maintained by the fovea. So you see this peak in this graph over here. This peak is the fovea, and is the fovea that contains the maximum number of cones, and the fovea is responsible for the sharpest visual acuity, which is measured by Snellen's chart. Temporal to the fovea is the optic nerve or the optic disc, and that is a blind spot, okay, which you see here. So this is about the form sense. So obviously, when we are focusing at a particular object, for example, a flower, the image of that flower is to be falling on the fovea, the point of maximum resolution or acuity. The next was the contrast sense. Contrast sense, see, you see these lines of different contrasts, you can make out the contrasts, right? So this is the contrast sense. In simple words, the ability to perceive sharp outlines of relatively small objects. The third is the color sense. Now, I showed you a wavelength spectra in the beginning where the rods and cones have different absorption spectra. So it's the same thing, okay? Now, appreciation of colors is a function of the cones and therefore happens only in photopic vision. In low light, the eyes are dark adapted and the objects appear gray. And there are four classes of photopigments found in human photoreceptors. So the short wavelength cones, that is the blue cones, the rods, and the medium and long wavelength cones, that is the red and the green cones. So there are two widely accepted theories of color vision, the Young-Helmholtz and the Herring theory. You have to know both these theories in details. They can come separately as your short notes. So what is the trichromatic or the Young's theory? It states that except red, blue, green, all other colors are perceived by combinations of these three colors. For example, perception of yellow is because of simultaneous stimulation of red and green receptors, but it fails to explain why people who confuse red-green color actually see yellow, because yellow is supposed to be made up of red plus green, right? So this theory fails to explain that. So for that, we have another theory, that is the Herring's theory or the opponent process theory. This assumes that there are three pairs of antagonistic receptor systems, that is red-green, blue-yellow, black-white, such that of this antagonistic receptor system, stimulation of one causes inhibition of the other, right? This explains visual processing at higher centers. Please note that these are the visual milestones. So the color vision is actually fully developed only by 5 to 6 months. Okay. Remember the formula of sixes. This will help you. So 6 weeks fixation, 6 months depth perception, and 6 years full visual acuity, complete visual acuity appears. Binocular vision. Now, color blindness. So the second part of today's lecture is color blindness. Now I'm going to try to explain the very basics of color blindness and what these terms protanopia and deuteranopia, these terms mean. Okay. Let's not go into too much of details. Please try to understand the terminology. So opsins on long and medium wavelength cone are encoded by X chromosome, right? By long and medium, when we say long and medium, what are we talking about? We're talking about red and green only, right? The short wavelength cones are not present on the X chromosome. So we are not talking about blue. That is why we keep talking about red-green color blindness. It is the most common type of color blindness. It is related to X chromosome. So it is a X-linked disease. Okay, and that is the majority of cases of color blindness. Being X-linked, it is more common in males, and females are usually the carriers of this disease, right? So, what do these terms protanopia and protanomaly, these things mean? Let us see. The word prot comes from long wavelength cones or the red cones, such that protanopia, prot means red wavelength cones, anopia means absence. So, in this anomaly, red cones are absent and these people are dichromats. That is, red cones are absent and the other two cones are present. That is what you mean by protanopia. So all protanopes are dichromats. Understood? Similarly, deuter. The word deuter means medium wavelength cones or the green cones. Here again, deuteranopia means green cones absent, means red and blue are present. So again, these are dichromats. I hope you have understood. Now, similarly, prot anomaly and deuter anomaly. So here, the word that has changed is anomaly. Anomaly means there is a defect, but not absence. So basically, these people, protonomally means red is defective, mind you, not absent. Deuteronomy means green cones are defective, mind you, not absent. So these people have a defect in any one of the cones, but all the three are present, albeit anomalous. So this is known as anomalous trichromats. Okay. So we have dichromats and we have anomalous trichromats. So dichromats have two types of cones and this is known as anopsia. Trichromats are known as anomaly. The two types of congenital red-green color blindness based on the affected cone are protons, which are 2%, and deuterans, which are 6%. Okay, so you see protons in which red is absent, uh, as seen by the red dotted line. You see that the curve of the graph is shifted more towards smaller, that is the blue side. Red is absent, so it is shifted towards the shorter wavelength side, whereas green is more on the longer wavelength side. Okay. These are the protons and these are the deuterons. So lastly, despite often being called red blind, green blind, protons and deuterons, that is the dichromats, have very similar types of color vision. So what an able-bodied person like you or me would see, that there is a red background with a green 2 written, deuters and proters are going to see it like this. Very similar. This is what real life is. And when we say red-green color blindness, it means that the red, green are indicative colors of confusion. It does not mean that red and green are absent. Please remember that. This is a very, very common mistake. And what does a tritanope see? A tritanope sees something like this. That is, there is confusion in all the three colors. Okay. So no more details on this. I'm sure details you can read in your books. So this was just to make the fundamentals of color vision clear. So you can find my presentation on the YouTube channel, and thank you for listening.